Okay, let's start then. So, well, thank you so much, Denis and Olivier, for the very kind invitation. It's a pleasure to be in Paris in May, always special, and it was such a... We are in Paris in June, actually, yeah. You said Paris in May when you invited us. That's why I said yes. Okay. But it's about timing, right? So you will understand in a moment why I am so interested in heterochrony, so basically being ahead or actually too late in time. Okay, so in the lab we are interested in the mechanisms that link human brain development and evolution. And today I will emphasize in particular on what we think is, not, I wouldn't say a type, but a set of mechanisms that relate to developmental timing and that may play a particular role in the frame of human brain evolution. So very quickly, I know this is the end of the day, but just to remind you a little bit of a few prominent, let's say, differences or specialization. There's nothing unique about the human brain, but the human brain, like any other brain, is specialized compared with the brain of other species. So it has increased cell number compared with, even with the chimpanzee. In particular, expansion of these neurons populating the upper layers that are born the latest during development, expanded areas of the prefrontal cortex, increased connectivity between the neurons, also larger size of the neurons, more synapses on these neurons, different functional properties. And basically, a lot of it has to do with changes, most likely anyway, in changes in the development of the brain. And as I think James in particular explained very well, there are various ways to think about, let's say, an Evo-Devo problem like this one. And what I want to entertain with you today is basically that prolonged development is actually at the core of many of the features that may characterize the human brain, and certainly is an interesting case study, so to speak, in understanding how developmental timing varies across species and across cell types. So, very briefly, when it comes to developmental timing, there are two prominent features that are considerably prolonged in the human compared with other species. One is neurogenesis, meaning that the generation of the neurons that will populate the 6 layers of the cortex, which are orthologous mostly between human and mouse, take about 7 days, 6 to 7 days in the mouse, while it takes more than 4 months in the human. And you may say this is trivial, but in fact it is thought to contribute in a significant way to the increase in the number of neurons, in particular that are generated at late step of neurogenesis, a little bit along the lines of the development of the ant that Claude told us about. But what I want to focus on in particular today is more this aspect, because I think that's one that's quite special about primate and human development, and that is the prolonged and delayed development of human cortical neurons. What do I mean by that? Well, I think this illustrates you well the point. The neurons, the pyramidal neurons that you can see here in the Golgi staining of a 1-month-old infant and in a 6-year-old infant are the same in number, but you can see how much more complex and larger they are. And if I had the space on the slide, I would show you that the brain is considerably increased in size between those two ages, and this is mostly because of dendritogenesis, axonogenesis, synaptogenesis, so basically making connections and making those neurons grow enough to become mature, but the whole process takes, so when you see here for dendrites, more than 6 years at least, and here you can see development of the dendritic spines, which are a proxy for the synapses in the prefrontal cortex, and I hope you can appreciate that they basically grow continuously up until the 2nd decade following birth, and you can see also how few synapses there seem to be, and this is correlated with EM data, at birth in cortical neurons. So what's interesting from a developmental biology perspective is that this phenomenon, as I told you, is taking place in neurons that are mostly autologous with one another. They display largely similar sets of genes over development, although we'll see there are interesting differences, of course. On the other hand, the temporal difference is completely out of scale, much more out of scale than all of what we heard from beautiful work of many people today, basically, meaning that while the process of neuronal development takes about 30 to 60 days to be generous in the mouse. Well, as I told you, it will take up to 15 years in the prefrontal cortex of the human. And interestingly, it's various stages, but in particular, those that concern neuronal development and connectivity that are at stake here. And this is leading us to the second, I think, interesting question that is more leaning towards, I would say, evolutionary biology and neurobiology, and which is what are the consequences of this prolonged development? Is it just so that in our lab, when we start an experiment with human neurons, it takes 1 year to get the result? Or are there actually advantages in vivo of having a prolonged development? And in fact, it's been proposed for many years that prolonging the development, in particular of synaptic connectivity, is a way to optimize neural circuits along prolonged periods, critical periods, you may say, of plasticity, so that they are better fit to their postnatal environment, to their kins, to their social, for their social interactions, and so on. So it's been proposed actually for, I would say, almost or more than a century by evolutionary biologists, that the fact that the brain of little humans takes so much time to develop is actually at the core of the optimization of enhanced performance of the human brain. But and now we get into the core of the questions I want to entertain with you today. On the one hand, well, up until recently, we didn't know anything about the mechanisms that could underlie this out-of-scale development, which, by the way, I call neoteny, and that's a little vocabulary thing that I've been arguing a lot, actually, with science editors, and we don't agree, but according to science editor, neoteny means developmental time. I would agree that neoteny is actually the retention of juvenile properties in an otherwise mature organism, meaning some things go fast, but other things go slow, or some things go slow, or things, yeah, so you're on my side, thank you. But nevertheless, if you read our papers, it's written the way the editor wanted. But so, and of course, that's really critical, and in a way is to contrast with what we heard from James and Mickey, for instance, about a sort of proportional scaling of developmental timing of various processes in a concerted fashion, while here it seems that there's a selection for something that is out of scale in terms of timing. And then the next question is, so we know very little about this mechanism, and then the second question is, what are actually the links between neuronal neoteny, as I call it, and brain function and dysfunction? Because this thing, which, as I told you, has been proposed for probably over a century by evolutionary biologists, well, like many other things, no offense, proposed over a century ago by evolutionary biologists, has not been tested rigorously, experimentally, so that we actually don't really know whether changing the timing of circuits has an impact on how they work. And I will not have much time to discuss that with you, but I think, let's say, it might be an interesting take-home message about the question of developmental timing, which is not only what is its origin, but also what are its consequences on basically body formation or brain formation, function, and in the end, fitness. Since, after all, we are interested in how these things evolve. I think questioning what are the consequences on fitness is interesting. And to us, this seems like the ideal model to do that. Okay, now, I will quickly go on explaining to you basically how we got into this question. It's not like we woke up one morning and said, let's study developmental timing in the cortex across species. It's actually data from the lab from a number of years ago now that sort of made us aware of the question and made us very curious about it. First piece of data is that when it comes to neurogenesis, as you can see here, you can get these cells. I'm sorry, the first piece of data from the lab is that you can get sequential generation of neurons that correspond more or less to the six types of neurons that will populate the 6 layers. You can do that in a dish from pluripotent stem cells. And interestingly, when you do this process from mouse CSF cells, well, it takes about a week. So pretty much like what's going on in vivo. And if you do it from human cells, it takes about 3 to 4 months, pretty much what's going on in vivo. So first, evidence that there may be a strong intrinsic component, not necessarily cell intrinsic, as James was alluding to, but at least about, I would say, the lineage of these cells with one another to control timing. And you can see here an example that I go upon quickly where Ira Espuni-Camacho, when she was a postdoc in the lab, co-cultured human cells and mouse cells. And after 2 weeks, all the mouse cells have become neurons. They've completed neurogenesis, while the human cells are only starting. Basically, only very few of them are only starting to become neurons. So I think a striking picture, let's say, of heterochrony of mouse and human cortical neurogenesis. But now let's get into the hardcore thing, I would say, neuronal development, neuronal maturation. So here, it's another set of experiments that we performed where we first transplanted mouse or human cortical neurons differentiated from pluripotent stem cells in a neonatal mouse cortex. And basically, and many others have done that before us actually with mouse embryonic cortex, if you put mouse cells into the mouse, they basically, those pyramidal neurons will develop, as you can see, in about a month. So basically following the timeline of the host, which is also the timeline of the transplanted cell. But if you change the species of the transplanted cell, and if you take human neurons, well, you can see now that to get a similar stage, you need to wait 6 to 9 months of development. So a timeline that is actually much closer to the timeline that I told you about of human brain development. So suggesting that there is a strong intrinsic component again in the development of these neurons within the mouse cortex. And since then, we and others have done this with, in a variety of ways and basically always come to the same conclusion that the timeline of development of the transplanted cells looks very much like the timeline that is found in vivo in the species of these transplanted cells and not in the species of the host, so the mouse. Now, what could be, now more at the molecular and cellular level, the mechanisms that would underlie this strongly intrinsic timing mechanism? So, again, I'm going to, in a way, revisit in a slightly different way what you heard about today, and I apologize for that, but basically you can think of it in two ways, and in that way it was beautifully introduced by Mickey and James. On the one hand, you can imagine that there are changes within the gene regulatory network, so that some genes serving as brake or accelerator will change the speed along which the gene regulatory network unfolds. Alternatively, you can think that there are mechanisms outside the gene regulatory network, or at least on the periphery, upstream or downstream, and that will, on the same GRN, act upon the speed of unfolding. And this is basically the first aspect that I want to discuss with you. So you heard already today about the potential role of proteostasis, of RNA turnover, and so on developmental timing. I want to discuss with you basically data that we obtain on the impact of metabolism, and in particular on mitochondria metabolism in that frame. And this is the work of two very brilliant people in the lab, Ryo Iwata, a postdoc who's still in the lab, soon on the job market, and Pierre Casimir, a very talented MD PhD student. So to test these hypotheses, the first thing that Ryo and Pierre said is, and that was, I'm insisting a bit on this because I think it was very important for us to find anything, and that is to set up a timing mechanism experimentally, meaning that if you look at a dish or in vivo at neurogenesis, since it is in mammals an asynchronous process, you cannot, like Claude, look at where you are to know what time it is. There's actually no way to know when you look at a neuron when it was born, so that you don't know how fast it went to the stage where it currently is. I hope everybody still follows. This is my Belgian Monty Python thing. But anyway, so to try, though, so to label the cells and know what time it is for these neurons, what Ryo in particular developed is this kind of smart genetic tagging. We're using a gene, NeuroD1, that is upregulated transiently at the time of neuronal birth, can be inserted in the neurons, and then a reporter is triggered at the time of birth date together with the addition of tamoxifen, so that you can genetically label the cells a little bit like you would do with EDU, except you can now use a variety of reporters. You can see here the neurons develop. I'm also using a legal UMAP to show you that at the transcriptome level as well, they follow a nice developmental timeline depending on how old they are. But now the key thing basically that Ryo and Pierre did is to look at mitochondria, because we reason that they may well be actually important in the process of neuronal development, as it have been shown in other systems, such as adult stem cells or cancer cells, for instance, in regulating fate transition. And so what they've done then is a slight modification of the system I just showed you, where instead of labeling only the neurons, they also labeled the mitochondria within these neurons with specific fusion proteins. And you can see here the result with mouse cells, mouse cortical pyramidal neurons, and you can see that very early on after birth, surprisingly actually, these neurons have very few mitochondria that are very fragmented, suggesting a high degree of fission. And then as you go on, they get larger and larger, and you get more and more mitochondria as the neurons develop. A way to put it is that mitochondria seem to develop as the neurons develop. You can see here the quantifications of those various morphological parameters. Now, if we do the same experiment in the human, then we get a second surprise, so to speak, which is that the same process occurs, but occurring to a very different timeline, meaning that, as you can see, the mitochondria grow in size and volume, but following a very prolonged timeline that I would like to call neotenic, in the sense that both neuronal development and mitochondria development seem to be prolonged in time in the human compared with the mouse cells. And so to us, this was a surprise, because in our naive view, we are not a group studying mitochondria at all, at that time anyway. Mitochondria were this thing that is sort of housekeeping and the same whatever you do, whether you look at different cells or conditions, and you can see here dramatically dynamic they can be across species and across time. Now, the second thing we did then is to ask whether this could have any functional consequences, focusing on the metabolism that we thought, as I told you, that we hypothesized could be one of the regulators of speed of gene regulatory network unfolding. And so to do this, again, we labeled the neurons genetically according to their birth date. We sorted them, and in this case, I'm summarizing you a lot of data here, but we measured their mitochondria oxidative activity and the various components of their electric transport chain using oxygraphy. And the result is actually strikingly similar to what I showed you before, so that over time, there is a striking increase in the activity of the mitochondria with the mouse cells, and a much slower increase in activity in the human cells. And when you look at comparable developmental time points, you can see that basically the overall levels are much lower in the human than in the mouse. So that's the second surprise, which then led us to the third question, which is what could be the origin of these differences. And I don't think we have found everything here in terms of answers, but there is at least one, and that is actually the metabolic rates, and I would say the relative metabolic rates when using glucose in human versus mouse neurons during development. So again, same setup of time neurons, but here what we did is to feed those neurons with an isotope of glucose and then do metabolomic assays to ask basically how mouse versus human developing neurons feed upon glucose. And I'm sorry for those who are still under stress disorder following metabolic courses from medical or biology or biochemistry, but I have to show you this. So glucose goes first to glycolysis to pyruvate, and then can be converted to lactate, so that the whole energy basically used is without mitochondria, or if it is used by mitochondria, it goes through the Krebs or TCA cycle, which then can feed upon OXPHOS. So really what we're testing here is what is upstream in part of OXPHOS, and what we found, again, I will spare you all the details, but is, I think, interesting, also in the light of what we heard today. On the one hand, we see that the metabolites that correspond to the Krebs cycle, that correspond to the use of pyruvate by mitochondria, are strongly decreased in the human cells, while conversely, the lactate production and the lactate accumulation are increased in the human cells compared with the mouse. So it's as if it's not like we can really conclude something about global metabolism, but more that because of the differential use of mitochondria in these cells, different use is made also of the metabolites, and that could have, of course, profound consequences of whatever is downstream of all these metabolites. We can discuss that more in the Q&A. But and interestingly, what this suggests in a way is that the human cells, I'm sorry, I'm going too fast, the human cells mostly use this pathway, while the mouse cells mostly use this pathway. And this pathway here is, you may have heard about it in a better known word as aerobic glycolysis. So basically the use of glucose metabolism despite the presence of oxygen all the way down to lactate, because the mitochondria are not there or not active enough, which is actually a process that was originally described when looking at cancer cells and suggesting that it could actually be involved in the pathogenesis of cancer. This is the famous Warburg effect that you may have heard of, that may or may not be linked actually to oncogenesis, but that seems to be the case with those young human neurons. Okay, now the reason why I'm slipping through cancer is that thanks to this hypothesis that metabolic changes basically may be at the core of cancer growth or metastasis and so on, well, a number of drugs or treatments have been identified by scientists in the cancer field to basically rewire the metabolism, so to basically make the cancer cells to stop to go through aerobic glycolysis and instead to use the mitochondria. And so we reasoned that perhaps using those treatments, but on the human neurons, would boost their mitochondria function, and we could then test directly whether all the correlations that I showed you actually mean some kind of causality between mitochondria metabolism on the one hand and neuronal developmental speed on the other. And here you can see the effect of some various treatments. I'm not going to go into the details, but basically, as expected, I would say it works. We can boost OXPHOS activity in the human neurons with those various treatments. It's important to note that if I had plotted the mouse OXPHOS activity, it would be around here. So meaning that the mitochondria themselves don't have the same competence to respond to the treatment as to become mouse-like. Nevertheless, they can respond by increasing their activity. And now we can finally look at what's the outcome on neuronal development. So we've looked at a variety of things. I'm going to go quickly here, even though if you think about it, these are the most important results in my view of what I show you today. So first of all, if you take cortical neurons at 60 days of development and depolarize them, well, most of them will remain inert. And they will remain inert because they don't express yet the right genes and proteins that make them electrically excitable like a mature neuron should be. So this is a readout of maturation. If we now pre-treat the cells for 10 to 15 days, then stop the treatment, and then challenge them, well, you can see that now close to 80% of the neurons become electrically excitable. So that's a first sign that maturation speed is increased, and we've looked at it in many other ways, including synapse number, synapse maturation and function. Last but not least, the growth itself of the neurons that is increased in speed following those treatments that boost mitochondria only. Meaning that if I summarize all of what I've shown you so far, well, on the one hand, there is a striking difference in mitochondria development over time during cortical neuron development. There are striking differences across species between at least human and mouse, so that humans are displaying fewer and metabolically less active mitochondria compared with the mouse. And finally, and importantly, if we enhance mitochondria metabolism in the human cells, we can speed up their development. Obviously, we don't speed them up all the way to the mouse, but we have calculated, looking at morphology in particular, that we speed them basically in a range of 5 to 6 weeks ahead of time, so something substantial. And experiments I didn't have time to show you also show that if we decrease mitochondria metabolism, for instance, by blocking the entrance of pyruvate into mitochondria, we can get the symmetrical effect, if you will, that is a decrease in the rate of development of the neurons in the mouse. We don't make them completely neotenic like the human, of course, but we decrease substantially the speed at which they can reach maturation. So I think there are two interesting things for us here, and maybe we'll discuss more about it later on, but on the one hand, there is this result that mitochondria metabolism itself seems to be causally linked to the developmental tempo of cortical neurons. I think it may have interesting implications for neoteny, for mitochondria diseases, other types of diseases as well. It's also coming perhaps as an interesting tool this is generating interesting tool to accelerate neuronal maturation in what we think may be a more physiological fashion than what has been done otherwise, since in a way what we do is basically look at a very basic compartment of the cell and make it basically more like a mouse neuron, which in principle should be normal. But obviously this will be very interesting to explore in the future what is the outcome of these neurons following this acceleration. I want to end up this part by showing you these data from a paper in cell metabolism. This is a compendium basically of many in vivo metabolic data of the human brain, and you can see basically those three graphs. Two of them are measurement, one of them is a calculation, and so you can see in blue basically the use of glucose without oxygen, so basically the aerobic the glycolysis. You can see in red the use of oxygen, so OXPHOS activity, if you will, and the difference between the two, aerobic glycolysis in black, and you can see basically how transiently it's increased during the first weeks or months following birth. So at a time that is strikingly similar to, I think, what we think we are modeling in our experimental system. So I think that's food for thought, that metabolism is clearly far away from being a housekeeping activity in the brain. I think we heard it beautifully also from the talk right before me, and I hope that this will be explored further and further, and I think we'll get more surprises. Now, with the remaining time, I'd like to switch gear in various meanings of the switching gear, actually, as you will see at the end of the talk. And I want to tell you also about the efforts that we've made to try to understand better whether there could be, after all, changes within the gene regulatory network that could control some aspect of developmental timing of cortical neurons. And this is the work, in particular, of Baptiste Libet-Philippot, a very talented postdoc, currently on the job market. What he focused on, basically, is actually a process or a type of mechanism that James also already introduced, the concept of new genes, of gene duplicates present only in the human or primate species. So this is just to remind you that gene duplication can be a source of genomic innovation. It's not been studied in a very systematic way because of the difficulties to study these genes. I'm not going to go into the details, but a number of years ago, we set up a large effort, in particular in collaboration with Franck Polleux at Columbia, and Iko Suzuki in the lab identified more than dozens of hominid-specific gene duplicates that are heavily expressed and dynamically expressed during cortical development. And among these, there's one here, SRGAP2, which encodes, which corresponds to a family of genes that I will tell you more about now in detail. So this family was actually discovered way before this by Franck Polleux on the one hand and Cécile Charrier when she was in his lab. And what they pulled out, basically, what they discovered is that there's a gene that's called SRGAP2, that's a Rho gap, mostly present at the level of excitatory synapses in the cortex, and the gene encoding that protein was duplicated several times in the human specifically, leading actually to genes that are truncated, that will encode truncated version of the protein that since then a lot of work suggests may act as an inhibitor of the ancestor protein. And the reason actually why this is thought so also at the functional level is because when Cécile and Franck overexpressed the human-specific truncated version in the mouse cortex, what they observed is a delayed maturation of the dendritic spines, those proxies of synapses that I showed you earlier. And they had a similar phenotype following loss of function of the ancestral gene, the SRGAP2 gene, that is present in the mouse. But so all of that was super interesting, but all of it was overexpressing a gene that has nothing to do in the mouse, since it only occurred in the human genome and during human evolution. So a lot of people, including Franck and I, we thought that at some point somebody has to take the task of testing the requirement of this human-specific gene for its potential physiological requirement in human cortex development. So that's what Baptiste did, and basically he followed a strategy where he knocked down SRGAP2C, so the human-specific gene, or SRGAP2A, the ancestral gene, using shRNAs that recognize specifically one or the other gene. You can see here, for instance, in human cortical neurons, the SRGAP2C product, so the product of the human-specific gene duplicate, and how it's basically washed away with those specific treatments, whereas SRGAP2A, as you can see, is not touched. In fact, as we will see later on, it may be slightly increased in content, in quantity. And then basically what we did, what Baptiste did together with other people in the lab, is to look at morphology, function, as well as dynamics of the transplanted human neurons, as we do routinely in the lab. I showed you a few images earlier, and ask how is the speed of development of these human neurons happening with and without the human-specific genes that we hypothesized could be involved in the neoteny of synapse development. And using dendritic spines as a readout, well, you can see here how dendritic spines develop in our model, in vivo, in the mouse. And you can see basically that it takes 18 months to reach their maximum. They correspond actually to mostly cortical neurons of the visual cortex, and that fits actually very nicely with the in vivo brain data. But you can see now if you remove SRGAP2C or B, the human-specific paralogs, you can see basically there is this dramatic acceleration in the generation of these spines. While conversely, if we remove the ancestral gene, you see that it's a mirror image. They seem to develop much slower. You can see the quantifications here. So we basically show here that in vivo, human cortical neurons require this particular gene to display the neotenic developmental timeline that they show following xenotransplantation. And this has functional consequences, so that, for instance, these neurons become then excitable and make synapses faster than the control. So this was a first interesting result, and the first proof of principle in a way that we identified here, a new component of the gene regulatory network, if you will, of cortical neuron development, only present in the human cells, and that seems to delay the maturation of their synapses. Now, the interesting twist that I will go too quickly upon, that I want to share with you, because I think it's food for thought, is that when we looked into the molecular mechanism, and this is all about synapses and so on, so I think this is too much for you at the end of the day, and also for me, but bear with me for a moment. We look basically at a number of synaptic proteins following the loss of SRGAP2A or of SRGAP2C, and we found basically two interesting things. So, and all these Western blots data are made from synaptosomes of human cortical neurons following manipulation of SRGAP2 ancestor or human-specific genes. So the first thing that we found is, as you can see here, that when we remove SRGAP2C, we get increased level of SRGAP2A at the level of the synapse. So this is nicely confirming, if you will, this hypothesis that we had from the onset, that the ancestral protein is somehow counteracted by the human-specific protein. And here is now the interesting twist and surprise that we came over, and which is that another synaptic protein called SYNGAP1, I'll tell you in a moment what it is actually strongly downregulated following the downregulation of the human-specific gene, while conversely, as you can see, it is actually upregulated following the downregulation of the ancestral protein. Now, for those of you who are still awake with me, and in particular those who know about epistasis and so on, well, I think you can hopefully see from there that SRGAP2C is reducing the levels of SRGAP2A, which itself reduces the level of SYNGAP1. And I don't have time to show you, but there's basically, that's what we reveal here, a reciprocal regulation between those two proteins that are present in all species, but regulated by SRGAP2C. Now, why is that? Why am I insisting on this? Well, because SYNGAP1 turns out, maybe I'm running out of time, or if I am, tell me. The bell will ring soon. Okay, so I'm getting quickly, but basically what we found, as you can imagine, is that SYNGAP1 is actually required also for the neoteny of the cortical neurons. But at the same time, it's also a gene that is required for normal human brain development. In fact, it leads to up to 1% of cases of intellectual deficiency and autism spectrum disorder. So we go then slowly, let's say, to the last aspect, which is that perhaps this is the first piece of evidence, and I'm going to go past this and go here, that the normal speed of synaptic development is regulated by two genes at least, two proteins here, SRGAP2A and SYNGAP1. We actually have data to show that this is also the case in mouse, for instance. So this is a conserved pacemaker, if you will, that is used probably in all mammals. But this pacemaker is out of balance in the human because of the presence of SRGAP2C. And this pacemaker is also out of balance when one copy of this SYNGAP1 gene is removed, and that is in the ID-ASD patients that display haploinsufficiency for this gene. So again, food for thought, but I think it's interesting to think about the fact that the rate at which cortical neurons develop may have functional consequences for human brain development, and there's potentially evolutionary consequences. So if I want to summarize, on the one hand, I showed you that very basic general mechanisms may take place in human neurons to control their developmental timing, such as mitochondria dynamic and metabolism, and then much more, I would say, local ones, such as this trio of protein that I showed you act together only at the synapse to control the rate of synaptic maturation. And if I may have one last minute, this is a way for me to introduce something that I know Denis will like, and which is, let's call it the Eddy Merckx model of developmental timing. So most of you, of course, don't know who Eddy Merckx is. He's probably the only Belgian character who was known at the time all over the world, a huge bike champion. Now, and he won here in Mexico in 1968 what is called the hour record. So he basically ran for an hour, and he ran for about 50 kilometers. And this record remained until 2003. And even at the time, he hired lawyers to defend his thing. So anyway, this is completely crazy. But look at his bike. He basically chose himself very carefully. You see the height of the way by which his legs are going to move oh my God, I missed the English vocabulary of biking now, but I guess these are the wheels, and these are, help me, James, how would you call that? Yeah, something like that, exactly. So he's been using the same for 1 hour, but that's usually actually not what we do. Only Eddy Merckx can do that. What we typically do is actually, on the one hand, to rely on this one, but then also relying on switching gears, so that in what seems to be about the same way to move the wheels, we actually set up a very different speed to reach the final place. And I think, again, if we go back to this question that James and Mickey in particular nicely introduced, of allochrony versus heterochrony, of proportional versus out-of-scale developmental timing, I hope you will remember now Eddy Merckx. And with that, I will thank all the people involved in the work, and I'm happy to take questions.